Okay, you can start.
Okay, thank you. So today I want to share with you broadly about research careers at an academic institution. My name is Christian. I'm an associate professor within the Division of Rheumatology at University of Washington in Seattle. And these are my disclosures. Yes, since this is an educational opportunity, is for you to be aware of, though none of those will affect anything that I would discuss with you today. So what I would like to discuss with you today is why and how to pursue an academic career in brief. I will then share with you a little bit about why one would like to study rheumatology and immunology and why that would be important. And then finally, I will share with you a brief story about a project that we are pursuing and give you some thoughts about how I think one could design a good project. So why to pursue an academic career, I think it's different for many people, but though there are some important factors that would align for most of us. And as you may recognize by now, I'm not from the US, but I'm from far away, and I have a different kind of path to an academic career, I think, than some of you. So I was born and raised in the southern part of Sweden, and Sweden is a beautiful country. If any of you ever had the opportunity, I would highly recommend you to come and visit. It's a country where you're always surrounded by the environment. And this is some of my pictures. And we're always surrounded by nature, water, and we have a rich culture also of, the Vikings and royal burial grounds and so forth. So there are lots to explore. And that could, of course, raise your curiosity to everything that is surrounding us, the biology and so forth. And even more so, I think, growing up on a small farm in the southern part of Sweden. We're always surrounded by wildlife, both the domesticated wildlife, but also the real wildlife, if you would, deer, elk, fox, and so forth. And this was something that brought curiosity to me early on in my life. I wanted to explore nature, the surrounding nature, wildlife. And so that was one of the main things that brought me into studying biology and chemistry, immunology, and so forth throughout my education. Another thing that may be more so particular to me in my upbringing is that, of course, being brought up on a farm, you're expected to work. And come spring every year, you were expected to go out on the field and pick up all the stones. And this is together with my twin brother and younger cousin, so that all the crops could be brought up. And every year, my father said the same thing, that once you become a professor, you don't have to work on the field anymore. So that was another kind of important aspect in my career, that you have the carrot kind of helping you be inspired and curious about the wildlife, but you also have something pushing you away from picking stones, I suppose. But this curiosity, I think, is key for any of you interested in a scientific career, that without curiosity, it's very hard to be inspired to put in the effort and the hard work required for a successful career. So it needs to be a deeply rooted curiosity to any topic that you decide to study. And a third topic that I wanted to touch upon, I suppose, regarding my background, and that relates to an important point too, is to start early on in your academic career or your scientific endeavors. And this is me about age 8. I went out into the fields and started to look at all of the insects and whatever I could find and characterize them. So here you see an ant, and you can see the wasp and so forth. And this is a small bug with an air supply. And me and my brother went out just to look at the animals and insects, and we drew some illustrations. We wrote where we found them, what time of the year, and so forth. And we also did some, tests for what they could or could not perform. We asked if they could climb, if they could bite, if they could sting, if they could fly, if they could swim. And of course, neither the ant or the wasp could swim, for an instance, which is recorded here. And we also had an access to a riverbed close to our home, which had limestone. And so there were lots of very interesting orchids as well as fossils in this riverbed. So we could always go out and just dig in the riverbed and find shark tooth or anything like that. So this is part of my upbringing, and being involved with and surrounded by nature and being inspired by that. And that was really what brought me into science to start with. We have a different school systems back home where we have elementary school for about 9 years, and then we do high school. And already in high school, you're expected to decide what kind of topics you would like to study. So I decided to go for a STEM-oriented high school, whereas other ones could go in for more, maybe how to become a mechanic and work with a car, or nursing school or anything like that. But already when you are 15 years old, you do decide to go into a STEM-related program. And then after that, you apply, when you then are 18, you can apply for your Master of Science education, which is 4 years at university. So the education is a little bit different, but the path forward is still similar, that you focus in on STEM-related topics if you want to pursue this career as a scientist. And thinking about this again, this early start of your career, I think that's a very important thing to keep in mind, and something I would recommend for all of you, wherever you are in your education. If you are considering a career that may or may not involve science, I would still encourage everyone to come to a lab, do some volunteering, get to know and see what would it be like to have a research career. And this could be in a clinical setting where you do some short reviews or epi work. It could be in a basic research lab where you learn to dissect mice or work with cell lines, or maybe even in a pharma company. But to be exposed to research early on in your career, I think would be very helpful. Not only for you to help decide whether this is for you or not, but it's also a very important opportunity for you to network and to get to know mentors that can help you in your future career. And also it really helps to build your CV for admission for graduate programs or other prominent schools. And that is certainly something we often consider when reviewing applicants' CVs, whether or not they have research background. And another thing that I often see when I review application or CVs or anything like that is that many students do have experience from many groups, from many research groups. And that could be a good thing. If you do not appreciate your current mentor, or if you don't receive any mentorship, I certainly would advise you to move to a different group. But it could also be good to have a broad experience and learn different techniques. However, there's also the balance that often the longer you stay within one group, the more opportunities you'll get to dive deeper into things and be part of publications and so forth that are also very important for your career. So it's a balance, and I think that's a personal decision as well, how well you work in a certain group. But I think that if you do well, I think it's important for you to build a long-term interaction with your current team and build towards independency and your own projects within that team. I am currently an associate professor, so I have been through a lot of different career steps, and I thought I would give you some just brief advice about how to think about an academic career. We've already discussed in quite some detail that curiosity is very important. Without curiosity, it's going to be very hard to find this drive and passion to stay in academia. Then you could as well go into industry or to any other field. But you need to be passionate and curious about something. And this is not only true in general, but in academia, you will, most likely at least, also receive lots of rejections for papers, for grant applications, and anything like that. And if you don't then have this curiosity and passion, you can be let down or become depressed even. And so it's important to have this deeply rooted curiosity and recognition that you are doing something that is very good. Another very important thing for an academic career is to find good mentors. And these need to be mentors that care for you and your career. You don't want to have mentors that are hiring you on as technicians, but you want mentors that are helping you in your career. You want mentors that are leaders in the field so they can help lead you into that position as well. Ideally, you would like to see a mentor that is experienced in mentoring and successfully leading other students into independent research careers. And ideally, you would also have mentors that publish good journals. This is going to also depend a little bit on what stage in your career you are. If you're volunteering, it doesn't really matter how well established the mentor is. As a PhD student, it is good if your mentor is well recognized, but it's not essential. However, as a postdoc, it is going to be almost instrumental that you are working with a well-recognized mentor that can help you develop good publications, as well as put you in touch with other good people that are working in this field and help you towards your independent career. And that's going to really be the one of the final points here that I would like to stress, that you need to network. And this is hard because many of us in academia or in research in particular are going to be introverts like myself. And networking does not come naturally as such, but it's something that we have to work with hard and that is essential for your career. And you need to network everywhere where you are, on your local workplace to find collaborators. You need to network on conferences to meet new potential collaborators, but also to meet with people that may eventually hire you or recommend you for certain positions, or that may be reviewing your grants and papers. And that's also, you need to be on social media and promote yourself and your work and your group. So there's a lot of networking and exposing yourself and your work that is going to be part of your academic career, more so the further you get in your career. And then a final point that I wanted to bring up, and there are of course many more things that you need to consider when it comes to your actual research, but another important thing in my mind is to always think about next step. Academic career is a very fast career where you only spend a couple of years in every position, and then you are expected to either transition into the next step or detransition away from an academic career and go and do something else. You have to always be on top of it and enable yourself to be ready for next promotion. And so once you are studying for your master degree, you need to start networking and find opportunities for your PhD position. And when you're a PhD, you need to network and find opportunities for a postdoc position and apply for grants and so forth. And all the time, you have to kind of consider, what is my next step and what do I need for that step? So it's a fairly stressful time initially in your academic career. Once you become an associate professor, at least at University of Washington, your timer, if you would, stops. There's no deadline for when you need to be promoted into full professor, but you can stay in this title for as many years as you want to. However, for all of the previous positions, there is a timer. And if you do not make it with sufficient publications, grants, and so forth for each and every one of those positions or promotions, then you may be having to depart from the institution. So there's a lot of pressure for you to promote. And that's something we are not going to discuss today, but that's another important aspect of this career, and why you also need to take classes, of course, in ethics and moral, and be sure that whatever you do is morally correct and that we don't engage in any fraudulent behavior in our research. So to quickly kind of try to emphasize these different topics and put it into perspective into how my career ended up. And I'm not necessarily saying that this follow the 101 step that I put together in the previous one, but it gives you an overview of what an academic career could look like. So up here, I have noted a couple of different positions. For some of you, this will be a similar kind of transition. And for some of you, there will be a intermittent transition between postdoc and assistant professor, where you do an acting position of any kind. And that could be very helpful. And we can get back to that as well. For many of you, you will spend about 4 to 6 years as a PhD student, and that's a very good time. And then here I just put the different institutions where I studied. And there are pros and cons with staying or transitioning. I had the opportunities to stay for 1 year as a postdoc at my previous institution. And that could be very helpful in finalizing some projects. You may have spent a lot of time in putting together your PhD thesis or your master thesis if you're in the step before, in that you may need some more time to finish everything up, as well as start applying for new opportunities to become a postdoc. And there are also some ideas whether to stay or not stay within the same institution after you finish your postdoc, and whether you should then go to a different institution and not. I find that you could very well stay within the same institution, and that may be beneficial in many ways, as long as you have a good opportunity to depart from your previous mentor and show independence. And that's going to be the main thinking. As soon as you become assistant professor, you would like to start to make sure that you have a distinct path of independency from your previous mentor. When it comes to publications, there's really no good guideline. There are some rough estimates of what you should have when you become an assistant professor, but this will also differ much depending on what area of research you have, and also what kind of publications you have, if they're top tier or if they're more mid-tier publications. I was fairly successful in the beginning in my career and got 11 publications, 6 of which were first authorship as a PhD student, and 2 of which were published in Blood, which is fairly good journal. And this rendered me the best PhD thesis at the time at our institution. It also gave me the opportunity during my PhD studies. It's a fairly small country. We're only 10 million people. And I was studying a rheumatic disease, and we had these national conferences. And it allowed me to present much of my work early on and network with these investigators. And that was really putting a good opportunity for long-term collaborations once I became independent. So even as a PhD student, I would strongly encourage networking and start building these established collaborations and friendships with your colleagues. But as you also see, even as a master's student, I was not able to publish anything. And if you can come in even earlier on and engage with volunteering and get publications early on, I think that's going to be very strong, make you help your CV quite a bit and allow for you to apply for good PhD studies. I was lucky to have a very good mentor during my second postdoc here in the US at University of Washington. He's well established, a leader in his field, and we were able to publish very well together. And that is not necessary, but it's of course very helpful for your career. We were lucky to publish a first authorship paper in Nature Medicine and in Journal of Experimental Medicine. And that helped me then to build further on these investigations when I became independent investigator, and allowed for me to get some NIH-based funding and continue my career as assistant professor. And then after 5 to 6 years, you're expected to transition. And I have now been associate professor for 2 years. And the main difference is, of course, that now we start to do a little bit more different things. And how you now would like to become meritorious is, of course, to continue to publish well, to continue to get good grants, but also more nationally and internationally engage with this different consortium, different companies, and be involved on a larger scale with different topics and areas. So to generalize, I think, and to put into perspective what you expect to do in the different academic careers, I think many of us, when we think about a scientist, we think about someone being in the lab. And if that is the case, then I'm not a scientist, because I have transitioned away from the lab. And I think that's kind of just a misconception of what science is. And there are so many different ways we can all contribute to science. And though I still love and miss lab work, it is nothing that I do too much of anymore. And that is something you're going to have to depart from. The further you get in your career, the less time you will actually have to dedicate to lab work. But it's a very important skill to have. And it's very important that you put in a lot of efforts early on to learn a broad range of different skills, whether it's going to be in basic science or in clinical science, that you learn very well the different techniques, because that's going to be helpful later when you would like to teach those skills to someone else. Instead of being in the lab, you're now going to dedicate more and more over the time to mentoring your group. I am currently really blessed with a fantastic group of people. We have currently 7 people in my team, varying from senior postdocs to recent undergraduate students. And it's just fantastic opportunity to mentor these people and to see them mentoring within the team. And that's something you're going to have to engage with much more the further you get in your career, to work with people. And that could again be another challenge for some of us who are more introvert, and something that we really need to work with as your career develops. Another thing is, of course, that initially you are more or less sustained by your mentor. But the further you get in your career, you're expected, of course, to sustain yourself and your own group. And the more and more time you need to sit in your office and read papers and write grants and hopefully get some funding. It's not an easy thing, but it's a skill that you will learn, and hopefully you'll be mentored to learn as well. And another fun part, I think, of advancing in your career too, is that you get to be involved with reviewing other people's grants and help kind of shape research that way. But you may also be involved more so in consulting companies. I'm currently working with many companies, and it's really rewarding to also be involved in trying to shape and develop drugs with smaller companies, or advising them in different disease settings and so forth. So you're really going to be going from being led by someone to become a leader in your field and lead others to the same thing. So it's just a matter of taking on leadership and becoming an expert. That's really what your career is about. So that was just a couple of brief kind of summaries, I suppose, of how I consider an academic career, and a few things to think about when it comes to curiosity, when it comes to this kind of different aspects of the different areas of your career. And I decided early on to start thinking about rheumatology. And just going back to my background, I suppose, after doing my first year in my master's in biomedicine, we had a summer break, and I reached out to almost all the professors that I knew or that I could find an email address to, and asked them if I could come and do a summer scholarship. And I only got one reply back, and it was about the complement system, and I had no idea whatsoever what complement was. But I thought, hey, this is the only one who wanted to work with me. I could as well go there and learn more. And I thought it was super fascinating. Nothing that I knew of or had any kind of connection to, but I think that's going to be true for many, many areas that you study. As soon as you get to know them, as soon as you start to studying them and learn more and more about them, you're going to have this curiosity just bubble up, if you would, and you're going to want to know more and more and more. Unfortunately, my then mentor, he shortly after that passed away, so I was transitioned to another group, now starting to study complement in rheumatic diseases. And from there, I got to know some of the leaders in Sweden who worked with this area. And through that consortium, I met with an invited speaker who came to discuss at one of our national meetings, and that happened to be Professor Keith Alcon, that was to become my later mentor. So I met him through networking and conferences, and then I applied to him and asked, hey, would you like to work with, could I come and work with you? So we applied for a grant together, and we were able to get that grant, and that allowed me to come to the US and work for a couple of years. And then I was supposed to return home. That was the policy of the grant, but for various reasons, I decided to stay in the US instead and accept a position here. And I think that was certainly the right thing. US is a wonderful place. UW is a really good place to study and to do biomedical sciences. And I happened to find my to-be wife here as well, and family here. So it was certainly a wonderful decision. So that's kind of my background and where I come from, and how I have done a scientific career or academic career, and something that I think could apply to many of you, independent on where you are currently. So what I study is rheumatology, and it's a super fascinating topic. in particular, since it's really difficult in my mind. It's still so unclear what is going on in these patients, and there are so many challenges still to be resolved. And here you can see a couple of famous people, if you would, that all are affected by rheumatic diseases. So this is a subspecialty or a specialty within internal medicine and or pediatric. It could be an autoimmune inflammatory disease. Many of you have probably heard of rheumatoid arthritis or lupus, maybe scleroderma, vasculitis. Gout is another one that is very common. But we have also other musculoskeletal diseases such as osteoarthritis that is very common, that also goes under the rheumatology specialty. So lots of different diseases, and I'm sure many of you know of individuals affected by these conditions. This is what rheumatic diseases could look like. You may be familiar with the butterfly rash that is very particular of lupus. This is going to be nephritis in lupus as well. We have gout crystals. You may have seen disformed joints from patients with arthritis and so forth. So it's really a disease that can affect any organ system, even though we primarily think of it as a joint disease. And this is a common problem in rheumatology. Osteoarthritis in particular is even more so frequent than diabetes, but even gout and rheumatoid arthritis have a couple of million people affected by it here in the US, and it's an ever-growing problem in our population. Another challenge is not only that we have an increase in the amount of people, but even though we currently are getting more and more advanced treatments, we're still not able to affect or prevent disease in many of these patients, with only 60 or 70% usually of patients responding to treatment. So there's a huge clinical need to help these patients do better, both in the clinical care as well as in research, trying to identify more and new ways and pathways to hinder disease in these patients. I wanted to briefly just give you one kind of case, one kind of insight into what it could look like to have a rheumatic disease. And then we'll going to go into more of the research that we're doing. So this is a young woman. This is just an imaginary description, by the way. But this is a young woman going here at UW. She's pre-med. She's 21, and she's now having had for the last couple of months some fatigue, pain in the joints, rash on the face, as you can see in the picture. But other than that, she has always been healthy. So these are new onset symptoms. She has pain and morning stiffness in her knees, wrists, and ankles, sharp pain in her chest, and she has also this doughy swelling of her ankles. She also seemed to have some foamy urine. She has anemia, for which she takes iron supplements, but otherwise she doesn't take any medicine, and she doesn't have any social or family history otherwise outside of these symptoms. Once she comes into the clinic, she takes her vitals, of course, and they are all fine, fairly normal. But at exam, you kind of verify her symptoms. She is looking tired. She has those red plaques on forehead, cheekbones, some moderate alopecia. She has painful joints, tender to touch, and there seems to be some swelling. But otherwise, her internal organs, as far as you can tell, with heart, lung, and GI, is normal. And then there's again a little bit of an edema of the ankles. And so then the question would be, and then they do, of course, a blood count as well, and she had still some anemia, but in a mild leukopenia, but nothing too complicated. So the overall question is kind of, what is this patient experiencing? Is there any kind of concern we should have with this diagnosis or involvement of anything? And so this young woman, unfortunately, is experiencing something we call lupus. And it's very common for it to start at this age. And one concern in particular for this woman is, of course, her urine being foamy, which could then indicate lupus nephritis, which is a very serious manifestation of this disease. And so lupus or systemic lupus erythematosus usually happens early on in life, with peak incidence when in individuals in their 20s or 30s. This is much more so common in female than male, with a 9 or 10-to-1 ratio. It does involve lots of different organ systems, and it could also be very variable, being either just mild with mild involvement of skin usually, and or life-threatening with involvement of the kidney, as we saw in the previous case, premature cardiovascular disease, neuropsychiatric lupus, and so forth. So it can really involve lots of different organs. And there are risk factors that are genetics, of course, sex factors. We know of environmental factors such as UV that could affect this. And we certainly do know that there is a racial prevalence or skewing, with African Americans being much more so, having a higher incidence of lupus as compared to white individuals, and much more so severe disease as well, including lupus nephritis, not least. It's hard to clinically diagnose lupus. We usually think of it as the kind of last resort disease if we can't think of anything else. It has lots of different clinical manifestations. Usually at diagnosis, the patient would experience fatigue and arthralgia. And then if you see it combined with another kind of manifestation of serology, let's say low blood counts, or any involvement of skin or kidney, that could merit diagnosis. We, of course, also have many other serological markers. You would often ask for an antibody test. You would look for anti-nuclear antibodies. Many of these patients will have antibodies targeting nucleic acids, such as double-stranded DNA or RNA components. And that is part of the clinical or the diagnostic criteria. But they could also have low levels of complement components and other things in the blood that we could measure. And this is one example of a rheumatic disease that we do study, and that is again very heterogeneous. And in the field, we're currently considering lupus not as one disease, but as many diseases, and probably that we need to engage with different treatment strategies depending on what kind of subtype of lupus these patients have. So it's a very challenging area to treat, since they come in and have so different clinical presentation and also underlying mechanism driving the disease. So it's very kind of rewarding to care for these patients, and also rewarding kind of to study this disease, because there's still so much we need to learn about these diseases. Another reason to go into rheumatology is that there is a huge demand for more rheumatologists. There is this idea that in a couple of years, we're going to just be lacking so many rheumatologists throughout the US. And so we certainly need everywhere, both clinicians and researchers doing work in rheumatology. So it's an open field for anyone interested in rheumatology. There will be opportunities to do good work, and to do good research. And I would, of course, argue that you should all come to UW and study rheumatology and immunology. UW is one of the best universities in the world when it comes to clinical medicine and immunology. Our research ranks very high as well when it comes to biomedical research. We're the top 5 university, or the 5th university in the world when it comes to the most innovative universities. We have lots of funding and even Nobel Prizes from our institution. So it's a very strong university overall, both when it comes to clinical care and when it comes to research. Our rheumatology fellowship is for 2 years, and we have 3 different pathways. One is a community practice if you're interested in clinical care in the neighborhood of Washington or the neighboring states. We also have one clinical scholar education where you can do some research, but primarily do clinical work. And then we have one pathway that is for 3 years, where you can really dedicate some time and learn to do some solid research. So if you're interested in a MD kind of career and do research, I would strongly recommend a physician scientist track. If you're interested in learning more about this, you could of course follow us on internet, or even send me an email, and I would be happy to put you in touch with anyone in our fellowship program. So now for the last part of the presentation, I thought I should also share a little bit with you what we are doing in my lab and why I think that is fun to do. And we are studying a couple of different things, but we are studying neutrophils, one of those innate immune cells that are just super fun, super challenging, but also involved in almost any disease you can think of. As soon as you have inflammation or infection, as soon as anything goes wrong, the neutrophil will go there and try to figure out what is wrong and cause some damage, unfortunately. So my lab has one part of our lab that studies basic biology when it comes to neutrophil functions. We study one fairly recent process in which the neutrophil just decides to explode throughout everything it has inside of it, even its own DNA. And it creates this kind of network or net-like structure that we call a net, neutrophil extracellular trap. This is very helpful when you want to trap and kill bacteria, but it seems like this DNA web-like structure is also very harmful in many conditions. It can cause thrombosis, it can facilitate metastasis, it can cause cardiovascular disease, and it seems to be very involved in autoimmunity and inflammation. So we try to understand, why does a neutrophil all of a sudden want to just explode and throw out all of this bad stuff? Can we regulate that? Can we remove them somehow? And how does these kind of neutrophils influence B cells to promote autoimmunity? So very basic aspects. But what I think is important and essential if you want to have a long-term career is to not only stay in the basic area of research, but to make your research translational, and even to anchor your research in things that are essential for patients. So we do a lot of work on biomarker discovery and development. We work with big pharmaceutical companies in identifying novel markers that can predict whether a patient will respond to a drug or not. And we also develop kind of markers to see whether we can stratify patients and learn more whether they will develop more or less severe disease long term, that can allow for early monitoring or maybe even more aggressive treatment to reduce the risk for those patients to develop long-term poor outcome. And in this particular study, we looked at something we call a neutrophil score, where we have measured different neutrophil biomarkers in solution. And we find that if these patients with rheumatoid arthritis lacked these neutrophil biomarkers, they were unlikely to respond to this particular drug. But if they had neutrophil activation, they were much more so likely to respond to drug. And this could be helpful then in moving towards what we call precision medicine or personalized medicine, where we only treat those patients that are benefiting from treatment, and thus reducing the time to recover and also reducing the side effects that these patients may otherwise experience. Another area we are very interested in, and that I will share a little bit more about, is the mitochondria. And that's also a fairly hot topic when it comes to immunometabolism and how energy kind of works in different immune cells. And we are particularly interested in mitochondria when it comes to not only thinking about mitochondria inside of cells, but a new emerging area of research is mitochondrial transfer. And that is something we're very interested in, how a mitochondria can be moving from one cell to another, transferring energy capacity and helping those cells that need more energy to get more mitochondria. And we'll go into a little bit more detail shortly and telling you what this picture actually is representing. Similarly, with the mitochondria, we also have a translational part of my group studying mitochondrial biomarkers, and we have developed novel technologies to study mitochondrial antibodies and so forth, similarly using them for patient stratification and looking at treatment responses. So what I wanted to say here is that I think it's important, in particular early on in your career, to be specialized, to have one area that you're fully pursuing. And I would argue that it's not necessary, it doesn't need to be a disease, but I think it's more important to study processes such as neutrophil activation or anything like that. And then you can apply that knowledge to different diseases. But it's important to become expert in one area to start with. And then when time permits and we have sufficient collaborators to help work with you, you can start expanding into more topics, but to keep it focused on one area and to be both basic and translational to make it relevant to patients is going to make it much more so easy for you to publish in good journals as well as to get good grants. So for this last part, I also wanted to mention one particular project that we study in our group that relates to mitochondria in a particular disease. And this relates to a disease in children and is called juvenile dermatomyositis. So this is an autoimmune rheumatic disease. It has a very low incidence, luckily, but we still have about 4,000 kids in the US with this particular disease. And similar to many autoimmune diseases, it's more prevalent in girls than in boys. And they often develop this disease about when they're 7 years old or so. As per the name, you may recognize that it involves both the skin and the muscle of these children. Usually they will see inflammation in the muscle and muscle weakness. And when it comes to the skin, they often have this inflammation and rashes throughout the body. One complicating factor that could occur in some of these children, ranging from maybe between 5 and 20% of the population, is that they can develop calcinosis. And this is the aggregation of calcium crystals in muscle or skin tissue. And it could form even some kind of exoskeleton and cause large crystals that may pierce your skin. They may cause harm and pain, but they may also pierce your skin and cause continuous infections and so forth. Really tedious and causing a lot of concern for many patients having them. Today, we barely know anything about why they occur or how to treat them. It's a hard, difficult disease to study since there are so few of these children, and even less so with calcinosis. So there's a huge need to understand better how is calcinosis occurring and what can we do to address it. And so when there is this need of understanding something, how I would address this as a researcher would be to start thinking about what is occurring locally in the tissue in these patients with calcinosis. So if we go closer and take a look at this muscle of this patient, what is happening in this patient? And what we see here is a badly injured muscle. But we also see inside something that looks like mitochondria. And of course, muscle cells need a lot of mitochondria because they need to produce a lot of energy. Here we have, this is by electron microscopy, by the way. Here we have a regular mitochondria that looks, by the eye at least, healthy. But you see most of the mitochondria have a lot of blackness or grayness inside of them. And these are mineralized or even calcified mitochondria. This is a fairly new concept and for sure had not been seen in juvenile dermatomyositis before. So almost all of these mitochondria in this poor child's muscle is calcified, crystallized. And that cannot be a good thing if your power plants are crystallized. However, are you going to get any energy from these mitochondria? And if you don't have any energy, how is the muscle going to work? Another important aspect, though, is that mitochondria is not only a source of energy, and that's usually what we think about when we think about mitochondria as a power plant, but they have a lot of other important aspects too. One being that they, due to their origin, also contain a lot of potential autoantigens that our body will react to. Many patients with lupus develop antibodies to cardiolipin, which is often associated with development of cardiovascular disease. Mitochondria also contain a lot of what we call danger-associated molecular patterns, things that will cause inflammation if they're seen by our immune system, including their N-formyl peptides. This is something like that will look very much so like a bacteria. The same with the mitochondrial DNA, looks very much so like a bacterial DNA. And that's why our immune system particularly think that mitochondria are a little bit strange, if you would, and they are very much so reacting to them when exposed to mitochondria. So mitochondria is not only producing energy, but they also have other important aspects that we need to consider when thinking about immunology. So all we had so far was this picture, what was going on in patients, and we could just imagine that most likely calcification would cause some kind of harm to the mitochondria. And what is important then is to take that clinical observation into your lab and try to replicate that. So we set up a method now to crystallize mitochondria within muscle cells. And doing so, we were able to demonstrate that yes, once you crystallize them, once you get calcium phosphate crystals into your mitochondria, they no longer are able to respirate and create energy. And that's measured here by a seahorse assay. And we could show that by many other ways too, but just to simplify it, it's important to bring it from your early clinical observation into the lab and verify it. Now you have a model that you can work with and try to intervene with and try to develop novel strategies to treat these patients with. We could also do some metabolomics and try to ask kind of what is going on in these mitochondria now as they're being crystallized, what is happening when it comes to energy production and so forth. And so this is kind of one of these omics approaches you can take to more broadly see what is happening in these patients or in these mitochondria. And you can do some pathway analysis using bioinformatics and see what is wrong with these mitochondria. And one thing that is happening when they become crystallized is that their mitochondrial electron transport chain is breaking down. And that is what is causing this lack or impairment of mitochondria, of ATP production. So this is all good, but this is a very tedious approach. We couldn't go and take biopsies from all patients and see whether their mitochondria are crystallized or not. That is not a convenient biomarker or way to approach this. So are there other ways we could address and kind of find an easy accessible marker of mitochondrial impairment? One of those potential markers would be a marker called growth and differentiation factor 15. So this is a soluble marker, which is always helpful. It's a blood sample you can take and measure this. It is particularly induced in skeletal muscle cells in response to respiratory chain dysfunction, so that's mitochondrial impairment. It's a myokine, meaning that it's derived from muscles, and it's a mitokine, meaning that it's induced upon mitochondrial dysfunction. So this could be a cool marker to see whether that could reflect maybe a mitochondrial impairment in muscle. And it has been shown to be involved in other myopathies or myositis, inflammatory myositis, but it has not been seen or analyzed in juvenile or adult dermatomyositis. So we thought this would be an excellent time to try if this marker of mitochondrial dysfunction in muscle cells would be also affected in patients with dermatomyositis. So we recruited two different cohorts, one from Chicago and one from Seattle. And you can see these are all young kids with an average age of about 10 years old. They've had the disease for quite some time. This is in months. And you see the majority of the patients are also female. And when it comes to calcinosis, you can see that here in Seattle, only 10% of the patients had a calcifying disease. So if we do measure these levels of this myokine in plasma samples from these patients, we do see that the levels of GDF15 is elevated in patients with juvenile dermatomyositis as compared to your healthy control children. And then here we are dividing it based on different myositis-specific antibodies that are, that's one way to phenotype these patients. So once you see that something is elevated, you want to understand whether it also is associated with different disease phenotypes. And we found that levels of GDF15 in these kids were strongly associated with disease activity, and both in muscle and in the total score for disease activity. And this association or correlation between GDF15 and disease activity score was very good with an R value of 0.71, which was much stronger than what we could find for creatinine kinase, which is the current gold standard to analyze muscle damage or muscle involvement in these kids. And doing a ROC curve analysis, we could similarly see that analyzing GDF15 had a better area under the curve value of 0.77 as compared to creatinine kinase with an area under the curve of 0.64, meaning that our new marker GDF15 is superior to current gold standard and should be used in a clinical setting to better identify patients with active disease and involvement of the muscle. So in the first picture, I showed you that mitochondria were damaged. The muscle cell was damaged as well. And we and others have shown that mitochondria are not always retained within cells, but they can also be released due to either active extrusion or due to when a cell is dying and the mitochondria is leaking out. And here we similarly in tissue from these patients could see that mitochondria were seen in the extracellular space outside of the muscle cell, just circulating around. And since we now saw that mitochondria is not only contained within cells, but also outside of the cell, we asked again if we could measure this in the peripheral blood using a simpler method. One way to do that is to measure extracellular mitochondrial DNA. And doing so, we found that a subpopulation of patients with juvenile dermatomyositis did have elevated levels of mitochondrial DNA in circulation. And that was more so elevated in patients with this calcifying disease, the ones that we expect to have more damage to the mitochondria. So this could be another potential biomarker to study mitochondrial release or mitochondrial dysfunction in these patients. And then for the final part that I wanted to mention was that mitochondria are not supposed to necessarily be outside of a cell. They are bacteria-like, and as such, our immune system may respond to them and produce antibodies towards mitochondria. This is very common. For instance, in patients with lupus, we often develop antibodies towards mitochondrial phospholipid called cardiolipin. And there is another condition called primary biliary cirrhosis, where one of the diagnostic criteria is antibodies towards mitochondria, and we call those AMA, antimitochondrial antibody M2. And then there are other conditions too, where you could get antimitochondrial antibodies. So this is a well-established phenomenon, but it is not known whether patients with dermatomyositis have any of these antibodies. So we set up a new method, and that's something our lab does frequently, and which is a good thing to do, to develop new technologies. And so we set up a method to isolate highly purified mitochondria. And we then can incubate these mitochondria with serum or plasma from patients with dermatomyositis. If they have antibodies towards these mitochondria, at least to the outer membrane of the mitochondria, these antibodies will bind, and we can then detect those antibodies using antibodies targeting human IgG. And these antibodies have then a fluorescent color on them, so we can detect them using flow cytometry. And this is just a flow cytometry plot. The further to the right you go, the more color it is, and that represents more antibody signal. And so doing that, asking whether we could find antibodies in JDM patients towards mitochondria, we found that indeed patients with calcinosis and juvenile dermatomyositis had elevated levels of these antibodies targeting mitochondria. And this is an odds ratio of more than 6 as compared to patients or healthy individuals. However, we also found that patients without calcinosis, some of them at least, had antibodies towards calcinosis, antibodies towards mitochondria. So we asked whether it could be that maybe these patients will develop calcinosis eventually, and that this could be a prognostic marker. And we tried that in a few patients that we had sampled longitudinally. And very well, what we found was that even before patients develop calcinosis, they do have elevated levels of these antibodies. So we could find a prognostic factor and an odds ratio of more than 11, that if you have these antibodies, you're highly likely to develop long-term these very disabling calcinosis in these children. And we have since done this used these antibodies in many other conditions and similarly found them to be prognostic of various disease developments. And this is just showing a Western blot now of this particular reactivity towards the mitochondrial component that we're currently pursuing by mass spectrometry to try to identify the exact identity of the reactivity. But so far, we have identified this marker as a highly clinically significant marker that can predict disease development or disease progression in these young kids. So to summarize, and some data that I didn't show, but what we have identified so far from these investigations is that in muscle of these patients with JDM, different traumas, including trauma to your muscle and repeated physical trauma, or hypoxia, will lead to accumulation of calcium within your muscle and within your mitochondria, and this will cause mitochondrial damage and harm. And this could be reflected by soluble levels of GDF-15. These damaged mitochondria will be released, and this will also cause less ATP production in the muscle cell and fatigue in the muscle. What I didn't show today is that these damaged mitochondria will also release some of their inflammatory components, including mitochondrial DNA and N-formulated methionine that will activate our immune system to cause local inflammation. And also show that these mitochondria are now immunogenic and can cause development of antibodies targeting these mitochondria. And these are then all useful to study and monitor disease activity and phenotype. And we're also currently pursuing more so in detail what is going on with this classification and trying to intervene with this process to prevent development of this disabling condition. And finally, this is the current team, as well as some of my previous mentees, as well as collaborators. And far to the right, you can also see my contact information if you have any questions on the content of this, or just want to reach out for any questions about career opportunities or anything like that. Please feel welcome to reach out, and I would be happy to talk further with you.
Thank you so much, Dr. Lood.
Thank you.